All right. Just wanted to remind you about the assignment coming up. It's due on Friday, posted in EduCat, in the top section of EduCat. And so it's the forest and fungi. There's a few readings that I posted, very short, from that book, The Hidden Life of Trees. And so if you pull up that document, it tells you what I'm expecting. All right. And you have a range of options. You only have to pick one idea from those readings and find a published paper that discusses that in a more scientific way, and then relate it back to your reading. Okay. I'm not expecting a huge thing, like a couple of paragraphs, nothing major. All right. I just want to see that you've given some thought to what is being talked about in these books. And then always, it's always a good idea to go into the literature and see if you can find things that support what the author is saying. Okay, questions? I'll need the link to the paper in your document so I can get access to the document, whatever one you picked, because I'll need to compare that to what you wrote. Okay. So Friday by 11 a.m. And then I believe this Friday they'll open another lab homework quiz thing for you to do at your leisure between Friday and then the following Wednesday. Okay. So I thought I could squeeze this one in here now and it wouldn't be too big of a thing. Questions? All right. So the plan for today is to continue on with fungi in the realm of how they can be symbiotic with other organisms besides plants, terrestrial plants. Let's see. So we were talking about lichens. And let me get this situated. So lichens are amazing because when you grow these organisms all by themselves, they look very different. The fungi usually are typical fungi. They have that hyphal growth form, the long strands. Algae and cyanobacteria are unicellular, and for the most part, or at least with this relationship. And then put them together, and you get quite a shift in the morphology. And I'm sure if you've been hiking around, or even in town, walking around in town, or hiking around here, if you start training your eye to finding lichens, you'll find them everywhere, which is very reassuring, actually. I'll explain why. But I think we left off thinking about this idea, that lower left image there, where you have the fungal hyphae in the green, they're stained green, and then the algae are mixed into that. And you can see how well protected the algae are in this case. And so that's definitely an advantage for the algae or the cyanobacteria. So the algae and cyanobacteria are busy doing photosynthesis. They have access to sunlight. They're able to produce sugar and feed that partly to the fungus. The fungus in return will be providing minerals because it's so good at degrading rock material and releasing minerals and other growth factors. And then we also found out in the not too distant past here that yeast, which are fungi, but they're unicellular type fungi, they can also be in the mix as well. And that has helped solve the mystery of why certain lichens, they seem identical in terms of the partners, but they behave differently. And then they looked a little bit more closely and saw that yeast were also in there too. And that's why they had a little bit different behavior in terms of their physiology. And so this was published by Purdue University and really has helped clarify some of the issues that they were having with studying lichens and some of the disparities that they were seeing. I highlighted it in blue. This is an excellent example of how things can be hidden right under our eyes and why it is crucial that we keep studying the microbial world. And so I thought that was very well put, and I'm always for people taking a second glance at microbes. They're super important. So we didn't know they existed until just a few years ago. So lots to learn yet when it comes to lichens. So a few things you can say. When you look at the species of algae that hang out in this lichen relationship, these three genera dominate. So I'd like you to be able to recognize these. It would probably be like a multiple choice question where I'd be asking you to recognize these. So Trabuxia, Pseudotrabuxia, and Trentifolia. Really, when you look at all the algae that have been studied in any detail, it's one of these genera. You'll find algal species from one of these genera. On the other hand, with cyanobacteria, really it's Nostoc, that genus. We don't really see much else yet. That doesn't mean they don't exist. We just haven't seen them. And so about 90% of the lichens that have been studied will have the Nostoc cyanobacteria or those three genera of algae making up that portion of the relationship. Yes. With lichens? Yeah, right. I know. So they all decided, they came to an agreement that they go by the fungal species. So when they name a lichen, it'll have the fungal species, the fungus that's involved. I don't know what they're going to do about the yeast. You know, I think they're going to stay with the tradition, the hyphal-like growth fungus. That's the one that they'll use for the name of the lichen. So fungi, there's a lot of different fungi that form this relationship, so much more abundant. They usually fall into the so-called ascomycete category of fungi, but there's others as well. There have been some lichens in two other classes of fungi. The fungus coming from, so at least three classes of fungi, we can see fungi capable of forming this lichen relationship. But the ascomycetes, at least right now, with all the discoveries they've made to date, they tend to win. There's more in that group of fungi. And there's roughly 13,000 counting that have been named, and they go by the fungal species name, and they actually document them. So as far as I know, no one's ever seen a lichen relationship form from scratch. I mean, that would be a long, I don't know how, finding that happen in nature, that would be, I don't know, that'd be very, very rare. And like I said, when you grow them up in the lab, they look very differently when you grow them separately. And so when you look at the lichens more carefully under the microscope, you tend to see that the fungal hyphae will wrap around some of the algae or the cyanobacteria. There's a couple of hypotheses for that. One is that it helps them disperse as a unit. And so that's what this image is trying to show. They're trying to show the Soredia. The Soredia are the unit of dispersal. And so that's the fancy name for these. In this case, it looks like algae cells that are wrapped around, have fungal hyphae wrapped around it. And then if you can imagine, a rain hitting the lichen body, and then the Soredia gets splashed away from that lichen to a nearby habitat, hopefully they'll grow. So they go as a unit, which makes sense, right? Because they have to find their partners. So if they have this already good to go, then like the body gets disturbed, hopefully it's going to be both the algae and the fungus that's going to get bumped into the habitat, whether it be rain or a deer walking over it or you walking over it. Okay, and then the ascospores idea, that's just the fungal reproduction going on. Okay, so that when the fungus decides to go through sexual reproduction, it will form ascospores. So you can see that the lichen body, they often refer to the body of the lichen. They give it a special term. They call it a thallus. Thallus. Um, it's they they refer to more than lichen thallus. But basically, when you look at a lichen, you can see that there's some differentiation going on. There's structures that kind of look like roots, but they're not really roots. They're not a plant. But they call them rhizines, and that connects them to the substrate, whether it's a rock or the tree trunk or whatever. And then you see it's more dense tissue-like growth of the fungi at the lower level. And then the looser growth in the middle is that the algae can be in there, they still get access to sunlight, and then there's a tighter growth a little bit above to a little bit more protection for the algae. And so there's definitely this structure and differentiation to the thallus or the lichen body. But the Serratia, I want you to know that term, please. The Serratia. You don't need to know Ceralium or Apothecium. I don't care about that. But Serratia, please know that, because that's a really important idea. It really makes good sense. It's a good survival strategy to have both partners going with any disturbance that gets them out into the habitat nearby. Again, a close-up of what it might look like under the microscope. So this is, again, like bluish green is all the fungal hyphae, and you can see that there's different, there's very dense toward the bottom of the fungal hyphae, and then it's looser in the middle. And you can see if you look closely, there's some algal cells mixed in there. And then at the very top, in this case, this particular image is showing all that red at the top. That's the fungal spores that are being produced through sexual reproduction. So those are ascospores being produced. So lichens are very durable, but they grow very slowly. Yes, slowly, just fungal. And it's those spores are the result of sexual reproduction. And so it doesn't really help the lichen partnership, it's just for the fungus. So the lichens grow really slowly. So you need to be appreciative of that. And Dr. Robertus and I took a lichen workshop several years ago here in the UP. A lichenologist from University of Michigan came up and gave an excellent workshop. And one of the things that he stressed, because there was like 45 of us in the workshop, and he said, now we're going to go to this habitat, we're going to collect lichens to bring it back to study it under the microscope, but you need to be respectful that they grow slowly. And so we had to take very small pieces, we had to watch where we walked. And so just keep that in mind when you're out hiking. They do grow slowly. They're a remarkable organism, but if you constantly step on them and just destroy them in that regard, that's not good. So they're very resilient in the idea that they can handle drought. They're very capable. And so they have strategies in place to allow them to dry out quickly, kind of go dormant, and long stretches of dormancy. And then when the water in the area becomes more abundant, they can activate. You can even do this if you ever find in the spring here some dry patches of lichens and they look dormant and it's warm enough out, wait for the weather to warm up. But if you take your water bottle and just drop it, water the lichen and hang out there for a while and meditate, you'll see the lichen start to green up and it'll start to look differently because it's becoming metabolically active. And then they can dry out again and go into dormancy again, over and over and over. And so they're so good at it. They often say it's like suspended animation. And so you can see them hanging out in Antarctic, or the Arctic, the Antarctica areas, and they're fine. They can handle that. And so they are really, really good at hanging out in areas of the world that are very extreme from the human perspective. And so you can find them all over the world, including, the really hot areas, the deserts, the super cold and very frequent, ice sheets, although ice is nothing but bare rock, high up on the mountains. But you can also see them on, human-made structures, roofs. I have moss, lichens and moss growing on the roof right now. But gravestones, the lower harbor breakwall. There's that orange growth on the concrete there. That's all lichens. I'll show you a picture of that one. And so they are famous for being primary colonizers in the sense that they can establish on rocks. Then because of the fungus having all those really interesting enzymes that can release minerals, and then you have decaying organic matter. And so really lichens can help build soil. It's slow, but it can happen. And so they are able to get into a habitat that's pretty fresh, where there's no obvious growth of plants, for example, and they can set up a situation that allows some more available minerals and the creation of soil, and then that helps other organisms come in and get established after the lichens have done a lot of their job. So Nostoc, one thing to know about that cyanobacteria is that it's good at fixing nitrogen. And I can't remember if we talked about that at all in this class yet, but fixing nitrogen is taking nitrogen gas from the atmosphere. So there's 80% roughly of nitrogen gas in our atmosphere, and converting it to ammonia, NH3. So N2, the dinitrogen, is not like plants can't make use of it. Even though plants need a ton of nitrogen in their diet, so to speak, they can't grab nitrogen gas and use it directly, unfortunately. The only organisms on the face of the earth that can do that are bacteria, including cyanobacteria and a handful of others. So there's no eukaryotic organs. Fungi can't do it. Fungi are awesome, but they can't do that. And plants can't do it, which is a shame because you have 80% dinitrogen and they can't touch it directly. So if you have a partner that can do this and you have high nitrogen, this is a good thing to have. And so that Nostoc is very good at fixing nitrogen and can hang out with the fungus and provide what they say a usable source of nitrogen, which is ammonia. So what they do is the bacteria will take dinitrogen gas. They have a very funky enzyme that's really good at breaking a triple bond. Dinitrogen has a triple bond that's incredibly difficult to cleave. So the certain bacteria have that enzyme. They cleave that, it turns into ammonia, and then right away that gets incorporated typically into amino acids by Nostoc, and in this case, the other partners in the lichen relationship. And so this is common strategies Nostoc now, especially with different lichens. Primarily, you're looking at regions that typically have low nitrogen or poor nitrogen for a good amount of the growing season in that area. So lichens can also hang out in, like I said, on cliffs and rocky areas, high altitude, Antarctica. And so here they're showing a cliff area in figure A, and then they show a close-up in figure B. I don't know how well you can see that from where you're sitting, but I think that at least the black layer you can see quite well. But all of this area here, that's actually lichen growing in rock material. And so talk about a survival strategy. If you are living, I think this is Antarctica, if you live there, you can bury yourself just a little bit down into the rock material. That's a pretty great way to get out of the elements and survive. And so pretty slick adaptation there. Lichens can also be very sensitive to air pollution. So that's why I said I'm glad that I do see a lot of lichens around here in Marquette, because that's pointing to the idea that we don't have that much air pollution They slow their growth and die in the presence of too much air pollution. They have no way to secrete it out of their body. So it accumulates and accumulates and accumulates, and they can't get rid of it. And then once the chlorophyll starts to get damaged, of course, it's going to be extremely challenging for the algae or the cyanobacteria to keep providing sugars. This one in particular, this atmospheric contaminant, sulfur dioxide, is one of the main killers of lichen, and that's something that humans, through industrial practices, we make a lot of sulfur dioxide that goes into the atmosphere. And so here you're looking at a graph of data that's very old, but it's kind of a landmark study where they were looking, I think this is over, in England, Newcastle upon Tyne. And so the city center is the 0 point on the diagram here, and then distance from the city. So you can see after you go out 10 miles from the center of the city, you finally start to see populations of lichens showing up. So wouldn't really want to live in that particular city, because what that's pointing to is that there's likely high levels of sulfur dioxide and other contaminants in the air. And of course, you can't just not breathe that. You don't have an option. So there was a student in BI 310 2 years ago that also did a study around here looking at a similar idea, a similar study, lichen density. And so she did the city center of Marquette and then worked her way out. And she did a very nice study looking at the even like where she found them on tree trunks, or were they north facing, south facing. So she really did some nice data collection. And here's some of the data, which was, some of it was statistically significant. North-south is referring to what part of the tree trunk, what facing side of the tree trunk. But basically, you can see that percent coverage of the different tree trunks she was looking at, definitely right in town. She was seeing them, and then all the way out. But you can see the trend as you go further out. You do see, higher percent coverage, which makes sense. And so, but I was very relieved to see that in Marquette itself, there's plenty of fungal species. So she did a really nice job. And this data is very nicely showing that pattern. Once you get out into the woods and further out, you just have better ways of spreading the fungi or the lichen as well in that regard. So lichens, when you look at their body, they look different from each other in a few key respects. So they've categorized them into 3 main categories, and I'd like you to know the name and descriptions of these categories. So crustose is one, crustose, crusty, think crusty. All right, so these are lichen bodies that are growing very tight to the substrate. They stay very low, very tight to the substrate. So they're flattened and don't grow, up off the substrate. And they actually look crusty. I'll show you some pictures. Foliose is given to the shape where when you look at the lichen body, it tends to look like a leaf design. So it's a little bit broader than crustose and leaf-like, but again, still fairly low to the substrate, okay? And they look like broad leaves, like a oak leaf or something like that. Fruticose is the last category. Fruticose are the lichens that can shoot up off the surface a bit, and they look bushy, branched. And so I'll show you some pictures. So those are the 3 main body types that we tend to see. And it depends on primarily who the fungus is for this shape and design. So here are some examples. So this one we have around here, the wolf lichen. So this is fruticose. It's very bushy or branch-like. It's coming up off the substrate. Looks like a tree trunk, perhaps. I have down in the notes why they called it a wolf lichen. It's not a very nice story, but basically they use the metabolites from the lichen to kill wolves back in the day. And so they were poisonous. So what they did, here's the nice invention, the lichen with its toxic vulpinic acid was mixed with ground glass and meat. Apparently a deadly combination. So they leave that out for some animal, usually the wolves or the foxes, to come along and eat, and then they die. So nowadays, it's much better reputation. That's why I put the genus name. Maybe we should refer to it as Laparia species, which is a much nicer sound to it. And you can extract some really lovely dyes from this lichen. And so it's being used heavily for that purpose now too. Hopefully in a very sustainable way, because you can't just, like I said, harvest a bunch of lichen and expect that area to recover anytime soon. It will not. So, and so here, this is more foliose. You can see how it's a little bit more broad. It's not so branchy looking. You have a little bit broader surface area, more leaf-like. Here's crustose, where it does actually look crusty, growing on rock. And you often see lots of different lichens together with different pigments. So even though the partner that's doing photosynthesis has a ton of chlorophyll, you might think, well, all lichens should be green, but that's not the case. We often see lichens with beautiful colors other than green, which in and of itself is beautiful, but here you're seeing lots of gorgeous orange and brown, green. And so very often when you see these brilliant other colors besides green, they think these pigments are being produced because the lichen is getting exposed to very high sunlight, and it's almost like sunscreen for them. They are able to use these pigments to deal with that very high light. Too much light can mess with, especially the photosynthetic machinery, cause damage. So it's kind of like us, getting a sunburn. You're actually, creating damage to your cells. And so this, kind of like our melanin pigment that allows us to deal with too much sunlight on our skin, these pigments are kind of analogous to that, helping the lichen deal with too much sunlight. Very, very beautiful. Here's a gravestone that's covered in lichens. This is from National Geographic. So once you start training your eye, after that workshop I took with Dr. Robertis, and we both remarked, the whole rest of the summer, and it's to this day, it's just amazing how that very intense one, what was it, 4-day workshop, and it really does train your eye to look for lichens. So I encourage you to do so when you're out and about and doing your hiking around, or even doing errands in town, hanging out in town. You'll see them if you look for them. Another foliose idea where it's more broad leaf design. Sometimes you'll see those cups, by the way. Those cups, that's part of the fungus trying to go through sexual reproduction. So those are, that's where the sexual spores will be produced. And here's another good example of foliose.
So there's certain like
Yeah, the British soldiers, have you seen those? They have the very defined cup-like. Yeah. In the cup, you mean?
Yeah.
I don't know. Yeah, the kind of cup shape is it's giving a lot of here. And if you cut into that tissue, look at it, that's where all so the sacs of tissue are called asci, and then the ascospores are inside. And so it's like a miniature fruiting body. It's like a miniature mushroom, the algae would be a little deeper down. So most of the top of the tissue is fungus, and then the spores, the fungal spores. And then the algae is there's probably, if you look at the number of algae in that growth area, probably much lower population than what we for yeah, they're really neat structures. So there's another folio creeping along the substrate, very leaf-like. So yeah, very beautiful colors, and especially under high sunlight conditions. So they're getting their water needs and their mineral needs met through the air, rain, fog, dew, and then the fungus' ability to break down the rock material for minerals. So they don't need like true roots like plants do. They don't have to do that strategy because they just get it from the atmosphere around them. The fungi is busy breaking down the rock material for minerals. So then they just absorb it over the whole body of the lichen. So this is the one that's down the break wall that's so well adapted to growing concrete there. Beautiful orange color. And so no matter where you go, you're going to find lichens hanging out. And even they're looking at in space what happens. Like, so they're looking at using here we're looking at simulated Mars conditions at the ISS and trying to study what the lichen was up to. So the one that's on the breakwall, that's the one that they chose for this particular study. So one of our former grad students, he did his undergrad here and then he stayed on to his master's, Ryan Rutherford, he did a whole master's thesis with Dr. Robertus on lichens in this area, including up in the Huron Mountains, which is north of here. So could go on and on and on about lichens, and I hope that you'll take the time to look for them when you're out hiking and walking around. They're everywhere. Okay, but so with fungi, in this slide set, we were looking at how they form relationships with plants, and both as endophytic organisms inside the plant body as well as mycorrhizal fungi. Then we started looking at the lichen relationship. So the lichen relationship has algae often as partners. So that's the next topic I want to get into, is looking at algae a little bit more carefully. And so that's the next slide set that should be posted in EDUCAT. So let's get into that. So let me go back here. Algae are really diverse organisms. They have a lot of diversity in their morphology. They have a lot of diversity on their sexual reproduction. So I'm not even going to touch that in the sexual reproduction because it's so diverse. So we're not going to look at that. But we are going to look at differences in morphology and some strategies they may have to adapt to the environment that they find themselves in. So, we do have a lot of algae around here. A lot of people are under the misconception that algae are like, oh, that's the ocean. And sure, algae are super abundant in the ocean, but they also are in the Great Lakes, and there's terrestrial algae too. And then many of you who are to growing plants hydroponically, you may find algae all over your rock wool, and so forth. So they are everywhere. In that regard, they're not just in the ocean. So they can be single celled, and usually you need a microscope to detect those. They can be colonial, which means you have a bunch of cells all put together in some kind of arrangement or mass. Those often you can see with the naked eye. And often they're surrounded by some kind of gelatinous sheath to hold them all together, so they won't float apart from each other or break apart too easily. So single cell, colonial, and then there's filamentous. Filamentous is pretty descriptive, very long strands of algae. show you some pictures of all of these. And then kelps are the ones that think about in terms of the big ones, because they can get up to 60 meters. And if you've ever had the experience of scuba diving or snorkeling through kelp forest, I've not done that yet. I'd love to do that. That'd be pretty amazing. They have their own little ecosystem in those kelp forests and the kelps themselves can be quite big, quite extensive, and they differentiate their body quite nicely where you have a holdfast. I would like you to know that terminology, please. That's pretty. We talk about kelp. Use these words rather than roots. We don't really say roots. So we say holdfast and stunning or stalk. Is the stem-like area, and then the blades, they don't really call them leaves, they call them blades. So I would like you to know the jargon for the kelp body, please. And then the gas bladders are distributed along the blades, and it's a good name for them because they're full of gas, and they help the blades stay upright enough so that they can get access to the water coming down into or the sun coming down into the water. Okay, so please know these 4 groups: single cell, colonial, filamentous, kelp, and then the kelp jargon terminology. And so you have these, pretty much all the elements fit into one of these categories in terms of how do they look. And I'll give you some examples, and I'll show you some pictures next of the different. So single-celled is the top left. And this one has some flagella, so it's nicely motile. And then here you see colonial, where you have a bunch of single cells all put together in a mass. Volvox is another colonial. This one's very interesting. It usually has all of these little dots, these small dots that you see in the picture. Those are cells. And then the bigger structures, that's part of their sexual reproduction, which I'm not going to get into. That's why they look different, but that's all the same organism. Volvox species. And then here you see some filamentous on the far right here, where they look long and thin. Here's more filamentous. So sometimes they can be a problem in lakes, causing overgrowth. We'll talk a little bit more about that. And then the beautiful kelp forest, where you can be dwarfed in the presence of these kelps. You can see all the gas bladders along the whole body of the kelp. Here's a close-up of some of the blades in the gas bladders. Okay. So morphology. Now, this is from your book, the newest classification. It's been shifting. Like I said, algae are very diverse. So it's been pretty challenging for studying them and trying to figure out their relationships to each other. So I am going to ask you to know some of these, which is on the next slide. So the next slide shows I've crossed off some of them, so you don't need to know those. But I would like you to know phylum Phorophyta, top row, then Chromophyta, Rhodophyta, Dinophyta, and then the bottom, Pterophyta. So we'll go through each of these, and I'll kind of give you an overview of their key characteristics. And this table is nice because it kind of gets you looking at the different, like how many species in general. And then this is actually how they start to tease them apart and figure out their relationship. They look at what kind of food, when they do photosynthesis and then they have extra, like how do they store it? Do they store it as starch? Do they store it as sugar, alcohol, oil, fat, manifold? So they actually look at that as a classification method. And then chlorophyll, all organisms, they all have chlorophyll A, so that's why it's not even showing up here in the footnote. All of them have chlorophyll A, but as you see, some will have chlorophyll B, some will have C, some will have D. So I would like you to know that, please. For example, a phylum That one has chlorophyll A and chlorophyll D, which is, the reason why I'd like you to know that is it helps you to better understand their adaptation to their particular habitat. And so chlorophyll D, for example, with red algae, red algae, they tend to be a little bit lower in the water column. And this particular pigment is really good at grabbing that wavelength of light that's penetrating that deep. And so these different chlorophylls is just really nice, a nice adaptation to their light source. What wavelength of light are they getting at? Flagella, I don't really use that, but some have flagella, some don't. All right, so single cell are, well, here, phylum Chlorophyta or Chlorophyta are the green ones. And so this one is probably the best well-studied algae. This is Chlamydomonas, and very interesting organism. You can see here what it looks like under the microscope, and the drawing. And so remember now, in the algae, we're very much eukaryotic, and so they have the nucleus and the other organelles. Okay, so Chlamydomonas, very well studied. It has 2 flagella, so it's very motile, very fun to look at under the microscope. On the other hand, we can have some overgrowth of green algae, in particular here in the Great Lakes, that can lead to harmful algal blooms. I would like you to know that phrase, please. It's a concern that's growing here in the Great Lakes. There's several universities, including Michigan Tech University, that is on the kind of watching for this and trying to figure out better ways to detect it earlier so that they can try to mitigate it. But it is a real problem. It's an overgrowth of very often algae, but sometimes there's cyanobacteria mixed in there too. And the issue is that they get out of balance with that ecosystem, and some of these algae can release toxins that can be dangerous to fish and other organisms. So harmful algal blooms, sometimes people just say HABs. We'll call them HABs. If you ever hear someone throwing around that acronym. Here's a very nice picture of colonial green. This is Volvox again. And so those are all single cells that have come together into this colonial morphology. And literally there's thousands of cells that kind of make up, it's a hollow sphere covered with this gelatinous material. And they all have flagella and they all beat in unison. So somehow they're communicating with each other to beat in unison so that they can, make a directed movement from point A to point B. And like I said, reproduction, the bigger cells that you're looking at, that's sexual reproduction going on. And they can do asexual. And again, I'm not going to get into details on reproduction because it's all over the board in terms of how they do it. It just kind of drives you crazy after a while. So filamentous, very nice images here of filamentous green algae. And so in lab, you'll be looking at these under the microscope. We'll have live samples for you to look at. Here's one that has more of a leaf-like look to it. They actually call it sea lettuce. The genus name is Ulva. And so this is a nice one to show more of the seaweed morphology, leaf-like. So here's another one. This one is Coleochaete. It's a green algae, and when you look at it pretty carefully, I mean, it looks like a leaf, like what we've been looking at with terrestrial plants. And those look like a bunch of parenchyma cells all stuck together to make the leaf tissue design. They also can find lignin-like compounds in the cell walls. And so the more they looked at it, the more they're like, huh. And so they are starting to question if this one might be the closest relative to what we see with terrestrial plants. And so the story is not yet complete. We don't know for sure. So there's a lot of debate and more study and more debate and some more study. So we'll see how this plays out. There's another one that's in high contention for this honor of being the closest relative, and it's this one, Cara, which is the common name is stonewort, which we have around here, especially in a lot of inland lakes and streams. You can pick this out. It's what it looks like if you make it out. Right? And so this one used to be, okay, so this one used to be in the green algae. And then enough scientists with enough data said, yeah, no, we should put it into its own file. So I would like you to be aware of that it is technically now put into the file of Carophyta. It's no longer classified as a green algae. So we have it all over the place here. So this one is also, they're saying, well, what about this one? This one might be the closest one. So between the Coelophaenae and the Cara, kind of up for grabs right now. But so we'll see what other data they're going to come up with here to show which way if they think it's really happening. And then, yeah, some scientists also say, you know what? It seems like it's really more closely related to mosses. So the story continues on. So green algae are certainly found around here. You can find them in our lakes and rivers, including the Great Lakes, but they're also terrestrial. And but so far, anyways, if you add them all up, most of them are aquatic in fresh water. But we can find them as part of lichens, as you know, and you can find them out on tree trunks, on the surface of soil. You can find them on snow, rocks, even polar bear and sloth fur has the green algae hanging out. So most scientists would agree with this. Terrestrial plants are believed to have evolved from green algae. But remember that Cara, Carophyta, there's still, that's starting to gain some strength. So, but for now, let's just say green algae or Cara would be acceptable. And so the reason that they say this is when you look at the phylum that the green algae belong to, they all have chlorophyll A and B, and that's what terrestrial plants have. They have chlorophyll A and B. The green algae, when they make extra sugar, they store it as starch. That's what terrestrial plants do. They have plastid, just like terrestrial plants. What else? We have cellulose, hemicellulose, and pectin being found in green algae in the cell wall. We see that the flagella of green algae are similar to the flagella we see on the plant sperm, the terrestrial plants. And green algae have phytochrome pigments that we learned about several weeks ago, where the phytochromes are being used to be able to tell the plants how many hours am I in the light, how many hours am I in the dark. And so this is not a complete list, but these are some of the more commonly pointed to reasons for why they think green algae might be the ancestor. Okay, so we'll continue on with green algae, and then we'll get into some of the other phyla on Wednesday. And then in lab this week, you'll be looking at algae under the microscope and macroscopically too.